Welcome to the Who, What, Why podcast. I'm your host, Jeff Schechtman. It's amazing how many times in the past few weeks I've read, how did we get here? Sometimes that refers to the public health and political response to the coronavirus. But it also sometimes refers to how it all began. 90 days ago, we were with friends and family celebrating the holiday season. Today, a global pandemic has shaken the world to its very core. The answer, simply stated, is that it all started with bats. Before pigs, before the wet markets in Wuhan, the evolution of bats might be referred to as patient zero. If this seems hard to believe or to understand, my guest, UC Berkeley researcher Kara Brook, is here to explain. Kara Brook is a postdoctoral fellow with UC Berkeley's Miller Institute for Basic Research. Her research focuses on understanding the role that bats play as reservoirs for highly virulent emerging diseases, including rabies and viruses. Kara is a graduate of Stanford and Princeton, and it is my pleasure to welcome Dr. Kara Brook to the Who, What, Why podcast. Kara, thanks so much for joining us.
Oh, thanks. It's a pleasure to be here.
As we look at this and past pandemics, things like SARS and Ebola, so much of it could be traced back to bats. Talk about that first.
Yeah, so bats as reservoirs for virulent viruses is, as you said, the focus of my research. We, graduate student and I, in my group last year, published a paper where we actually were able to demonstrate that bat-borne zoonotic viruses, zoonotic means passing from an animal reservoir to a human host, and that bat-borne zoonoses are the most virulent of all mammalian zoonoses, so meaning they cause the highest case fatality rates upon emergence into the human population. And we think that the reason for this is related to the evolution of flight. So bats are the only flying mammal, and flight as a physiological process is more metabolically expensive than any form of terrestrial locomotion. So a human at full speed running will raise their baseline metabolic rate 2- or 3-fold. A rodent will raise their metabolic rate up to 7-fold, but a bat in flight will elevate its BMR, basal metabolic rate, up to 15-fold when flying. And typically we expect that metabolic activity trades off with longevity. So species that have really high metabolic rates tend to be much shorter lived. So rodents are a perfect example of that. But sort of flying in the face of that prediction, bats are the longest lived for their body size of any mammalian taxon. So the longest living bat that's ever been recorded, the Brandt's bat, has been found to live up to 40 years in the wild. And a rodent of the same size, it's only a couple of grams, would live up to 2 years at most. And so what we think is going on is that bats, in order for flight to even have become evolutionarily possible to begin with, bats needed to evolve hyper-efficient cellular mechanisms by which to reduce the oxidative stress that's incurred from the general process of metabolism. So metabolism accrues oxygen-free radicals as a byproduct, which then get oxidized, and the damage gets mitigated and repaired by cellular functioning. But when that damage accumulates and outpaces the body's ability to reduce it, that's what we call oxidative stress. And generally, over time, the accumulation of oxidative stressors leads to senescence, so leads to the inability of our tissues and organs to continue functioning, and that's what we think of as aging. And so bats, it seems, as a mechanism to support their ability to fly, which is so metabolically expensive, have evolved these really efficient DNA damage and repair pathways and oxidative stress mitigation pathways at the cellular level that enable them to withstand this highly physiologically expensive activity. And then as a consequence of that, bats have evolved this extraordinary longevity because their cells are so resilient to these stressors. And more recently, we've started to think that this also might play a role in their resilience to viral infection. So viruses in a cell will cause oxidative damage directly to that cell. And then in addition, a viral infection will lead to the recruitment of immune cells to the site of that infection, causing widespread inflammation. And that is also an oxidative stressor. And so it seems that because of flight, bats have evolved these efficient pathways to mitigate that damage, but then that's also allowed them to be long-lived and to be really resilient to virus. So they can host these viruses such as Ebola or Marburg or SARS or MERS without actually experiencing disease, without getting sick.
If we can, let's back up a little bit and talk about how the bats could infect other mammals even before they get to humans, and how that transfer then to humans takes place.
Yeah, so typically in disease ecology, which is my field, we do a lot of epidemiological modeling. And when we model the process of transmission, so passing a pathogen such as a virus from one host to another, we estimate this transmission rate, which we usually use the Greek letter beta to describe. And encapsulated in that transmission rate, beta, is both the infectiousness of the first host that's passing the infection, the susceptibility of the host that's receiving the infection, and then the contact rates between them. And so any of those three components of beta, modulating any of those three is going to affect the probability of transmission taking place. And so with respect to infectiousness, research is suggesting that there's a certain seasonality to bat infectiousness. They appear to shed more virus during periods in which they're immunocompromised or otherwise physiologically stressed. So during the reproductive period, and then also during nutritionally dry periods. Many of these infections come from fruit bats, and so there's a distinctive seasonality to the fruit availability in the country that these bats, whichever viral emergence you happen to be focusing on. So that's infectiousness. Susceptibility is going to vary by host to host. There might be seasonal factors related to that as well. There might also be certain individuals are more immunocompromised. And then also we've discovered that there's an inverse correlation between phylogenetic distance between two hosts and the probability of zoonosis. And so it seems that viruses that have evolved for a long time in a bat are more likely to transmit to an organism that is closely related to a bat. And so bats and humans are quite phylogenetically distant from one another, though they are both still mammals, but they're about as far apart mammalian orders as you could find. And so often what we see in this transmission process is that viruses will first spill over from bat to what we call an intermediate host or a secondary host. And in the case of SARS, that was a civet cat. In the case of MERS, that was a camel. In the case of Ebola and Marburg, we sometimes see non-human primates. In the case of Hendra and Nipah henipaviruses, Hendra it's horses, Nipah it's pigs, and then from that organism to a human. And so that acts as sort of a secondary bridge host that bridges that phylogenetic distance between the bat and the human. There are some bat-borne pathogens that can spill over directly. Nipah virus, for instance, in Bangladesh will spill over directly every year from bat to human, but they're sort of interesting, unique transmission pathways that enable that host jump. So in the case of Nipah virus in Bangladesh, the virus is shed in bat urine. And in Malaysia, we saw Nipah emerge first through a pig host. So it went from bat, where they shed virus in their urine. It landed in pig paddocks. Pigs obtained the virus, and then abattoir workers became infected from handling the pigs. But in the case of Bangladesh, people actually consume date palm sap there, and bats roost in these date palms. And so when virus is shed in bat urine into the sap and collected in these vessels that people use to collect sap for drinking, that can be infected with virus and give this very high dose that is able to make that evolutionary jump. And so a place like the wet markets that we see in southern and central China bring together a lot of these organisms that are not typically found in close proximity. And that brings us to that third component of data, the transmission term, that contact rate. And so something like a wet market is going to really increase the contact rates between distantly related hosts and make the probability of emergence between them much more likely. So in the case of SARS-CoV-2, which is the virus that causes COVID-19, we've not yet categorically demonstrated a clear secondary host. There's been some discussion of pangolins, some discussion of pigs, but no clear evidence whether there was a secondary host, and if so, what that host might have been. But you can imagine that bringing live bats in captivity in close proximity to something like a pig or something like a pangolin that they might otherwise not encounter in the wild is going to greatly facilitate the probability of a virus transmitting from one host to another and then on to the human.
And once it transfers to humans, talk a little bit about how those characteristics of the bat relate to humans and the way the human immune system responds to it, almost in overdrive, which is part of what's killing people right now.
Yeah, so because bats are so resilient to viral infection, this hypothesis related to flight, I've done some work both in cell culture and also some theoretical modeling work that suggests that viruses evolve different evolutionary optimum. So typically we think that viruses will evolve to balance their rates of transmission and the virulence incurred on their host, virulence being damage to the host. So what we think of as disease. And so typically a virus is not going to want to hurt its host because a virus's evolutionary goal is to transmit to as many new hosts as possible to maximize its rate of between host transmission. And we refer to that as R0, the basic reproduction number for a pathogen. And R0 is both the compilation of the number of new transmissions divided by the loss of transmissions through the loss of infected hosts. And that can take place through a host recovering from infection or also from dying. And so if a virus replicates very fast, it might improve its ability to transmit to new hosts, but at the same time, it might be killing its first host much more quickly. And there's a trade-off between those two, where at some point, even though it's replicating fast, reaching high viral loads that are likely to infect others, that host is going to be dying too quickly for the pathogen to be able to spread any more quickly than it was before. And so we call that its optimal virulence, when it's balanced that equation and maximized its R0. And in the case of bats, since they are so resilient to these viral infections, we think that essentially the optimal R0 for this pathogen involves a very high within-host rate of virus spread, because that viral replication in the bat host is not actually damaging the bat. So the virus, the sort of evolutionary ceiling on that trade-off is lifted, and a virus can evolve to either replicate or overcome host defenses much more quickly to enable it to reach higher viral loads in its host. And when that emerges into a host that is not a bat, we expect that would cause extreme pathology, because our immune systems are not as equipped to deal with those infections as others. And so one good example of that is that several species of Old World fruit bat have been demonstrated to have constitutively expressed interferon alpha. Interferon is an antiviral cytokine, which is a signaling protein. And typically what happens when a virus invades a cell, the viral RNA in that cell, the cell perceives that the virus has infected it and secretes interferon to initiate this downstream signaling cascade that alerts neighboring cells to the presence of infection and induces this antiviral state where those neighboring cells secrete a series of host factors and basically go into lockdown and trap the infection from spreading any more quickly. And so in a human host, we couldn't have a constitutively expressed antiviral response because it would induce widespread inflammation. But we think that that's due to their evolutionary adaptations related to flight are able to sustain these otherwise inflammatory pathways. And so we can imagine that if a virus co-evolves with a bat host that has these really robust antiviral immune defenses, it's going to evolve ways to circumvent those processes and still maintain infection within that host. But then when it emerges into something like a human that lacks those pathways, it ends up being too virulent, showing maladaptive virulence and killing that host much faster. What I should say is that in the case of this SARS-CoV-2 that causes COVID-19, this virus actually is very well adapted to its human host. It's not as virulent as SARS or MERS, for example, both of which had higher case fatality rates than the human host, but were much less transmissible. So in the case of this virus, we think that we've seen a bit of evolution in the human host that now means that this virus is well equipped to spread from human to human without killing those people too quickly before it achieves a new infection.
To what extent can understanding all of this help us in the treatment of the virus?
Yeah, it would be difficult to try to induce the same sort of immune responses that we see in bats in human hosts. As I mentioned, we're not equipped to sustain that kind of inflammation. So it's not really an effective pathway for therapeutics. But understanding the transmission dynamics in the reservoir host is absolutely essential to avoiding these spillover events in the future.
One of the things that also goes on is what's going on with bats today and the loss of habitat throughout the world and the impact that that's having.
Yeah, so as I mentioned, transmission from a bat host to a human or to a secondary host is dependent on this term beta that involves both infectiousness, susceptibility, and these rates of contact. And when we encroach on wild habitat where these animals live, we're elevating that contact rate and making it much more likely that we are going to encounter pathogens that we would not otherwise. And the same goes for our domestic wildlife, such as pigs, that are often these secondary amplifying hosts. At the same time, our work, I'm part of a collaborative network called Bat1 Health. We work in Australia studying Hendra virus, in Bangladesh studying Nipah virus, and then in Madagascar and Ghana studying bat-borne hennepaviruses in the same family. And what we've been able to demonstrate is that bats in intact habitats under good nutritional condition actually shed less virus in their urine during these annual pulses that take place during the nutritionally poor periods of the year. So bats that are in better condition pose less of a zoonotic threat. And so increased anthropogenic impacts on wild animal habitat elevate the stress levels in these wild organisms and then also compromise their immunity, which can lead both to heightened contact rates between bats and human hosts and to heightened infectiousness in the bat host through the process of viral shedding.
Tell us a little bit about where this research is going on right now and who are the people that are funding and responsible for it.
So as I mentioned, we work, our Bat1 Health team works in 4 global field sites. My work is focused in Madagascar. I study 3 species of endemic Old World fruit bat on the island of Madagascar and work very closely with Institute Pasteur of Madagascar, and in particular, Malagasy biologist Hafalina Christian Ranavusan. The field team in Australia is led by Dr. Raina Plowright of Montana State University, Dr. Ali Peel of Griffith University in Australia, and a number of hardworking graduate students and postdocs who work with them. There's a team that is joint with Johns Hopkins University and led by Dr. Emily Gurley that works in Bangladesh. And then our team in Ghana involves a number of Ghanaian scientists joint with a team at Cambridge University as well.
To what extent can all of this be anticipated looking forward, that we are going to see future pandemics not unlike this also emanating from bats?
I think that bats certainly serve as a source of highly virulent, dangerous pathogens, and the more impacts on wild fruit bat and insectivorous bat habitat that we're going to see into the future are going to be paired with future zoonoses. I mean, already in the past 20 years, we've seen outbreaks of SARS and MERS, and now SARS-CoV-2 coronaviruses. In addition, Ebola and Marburg, filoviruses, Hendra and Nipah henipaviruses, where we see human cases every year. So this is a recurring problem, and zoonotic pathogens pose some of the greatest threats because we are not able to control the infection through human public health. The Black Death was so globally destructive because a lot of the public health measures that we're taking to combat COVID-19 were ineffective because there was a rodent reservoir for that pathogen. So regardless of how we vaccinate or control a pathogen within the human population, if it has a zoonotic source, it's going to become a recurring threat. And in the case of coronaviruses, they're highly, highly diverse in bat populations globally. And several have demonstrated their ability to enter human cells, to bind this human cell receptor ACE2 and become infectious in the human population. So I think that it's really important that we be aware of the potential threats posed by these bat populations, but also understand that processes and patterns of exploitation actually exacerbate this problem, and that by working together with conservation biologists and public health practitioners, we can achieve win-wins for both wildlife conservation and human public health.
And what is the cutting edge of the research right now? What is it that you and your colleagues are still trying to uncover and understand about these processes that we've been talking about?
So bat immunology is a very young and up-and-coming field. I mentioned some of the pathways, this interferon response, but there's a number of unique host virus-specific evolutionary adaptations taken by different bat species with respect to different viruses. Also, to an extent, we sort of off the cuff tend to say that bats don't get sick from these viral infections, but in most cases of reservoir hosts, we've eventually been able to demonstrate that there is some sort of pathology. So a great example is SIV, simian immunodeficiency virus, which infects chimpanzees for a number of years. It was cited in the scientific literature that SIV was totally non-pathogenic, but took a 40-year data set coming out of Jane Goodall's work to demonstrate that there actually are more survival effects on the offspring of infected mothers. And so demonstrating fitness effects of these pathogens on the bat hosts, understanding exactly how they cope with them and what the sort of trade-offs are, might give us an opportunity to understand if any of these unique bat defenses are something that could be leveraged for something like immunotherapy in a human.
Kara Brook, I thank you so much for spending time with us today.
Yeah, thanks. It was a pleasure.
Thank you. And thank you for listening and for joining us here on Radio Who, What, Why. I hope you join us next week for another Radio Who, What, Why podcast. I'm Jeff Sheckman. If you like this podcast, please feel free to share and help others find it by rating and reviewing it on iTunes. You can also support this podcast and all the work we do by going to whowhatwhy.org/donate.